the enzyme that's involved in the carboxylation reaction of taking inorganic carbon dioxide and adding it to the 5-carbon sugar ribulose 1,5-bisphosphate to create 2 molecules of phosphoglyceric acid, the first step in the Calvin cycle, and certainly one of the most important enzymes on Earth in terms of converting inorganic carbon, which is not usable as an energy source for organisms, into organic carbon that is usable by all organisms on Earth. Okay, so we said that Rubisco had 2 main problems. It has a slow catalytic cycle. And it has very low affinity for CO2. It doesn't bind CO2 very effectively. And when we left off at the end of the lecture on Tuesday, the last thing we were talking about is a third problem, is that molecular oxygen is a competitive inhibitor. So who remembers what a competitive inhibitor is? What's the characteristic of a competitive inhibitor, Hemi? Yeah, that's right. It binds at the same site. And it's not hard to understand that because carbon dioxide looks like that and oxygen looks like that. So it's not completely crazy to think that those two may bind at the same site. So what that means is binding of oxygen prevents binding of carbon dioxide. Right? And that by itself is bad. But it's not just that oxygen is a competitive inhibitor. It actually goes through a chemical reaction. So there's a competing reaction, the oxygenation reaction, in which oxygen is added into RUBP rather than CO2, which produces 1 molecule of PGA, the normal 3-carbon product, but it also produces 1 molecule of phosphoglycolate. And as I mentioned at the end of lecture on Tuesday, the problem is that glycolate is toxic and cells need to do something with it. Okay, so we need to think about how important is this oxygenation reaction and what are the consequences as far as the plant is concerned. So let's first of all think about binding affinity. For oxygen and CO2. So CO2, the binding affinity is about 80 times the binding affinity for oxygen. So CO2 binds quite a bit more effectively than oxygen. But the problem is that dissolved in solution, the concentration of oxygen, let's make sure I get this right. Yeah, is 25 times the concentration of CO2. So although the binding affinity is much higher for CO2 because there's a lot more oxygen, it means that 1 out of 4 times that something binds, it's going to be an oxygen rather than a CO2. So under normal circumstances, as much as 25% of the RUBP that's produced goes in this direction rather than in this direction. So there's a potential for a lot of carbon being diverted into a pathway which is not the desirable outcome of this reaction. We need to think about how plants deal with it. And the way this is dealt with is through a process that's referred to as photorespiration. The respiration part of it comes from the fact that there's an uptake of oxygen, and as we'll see in just a minute, there's also a release of carbon dioxide. So that behaves like respiration. But it's a process that only happens in the light. It's completely light dependent because the regeneration of RUBP depends upon the light and the Calvin cycle. All right? So it's a light-dependent respiration. Don't think of it as anything directly related to the respiratory pathways that we're going to be talking about in a couple of lectures. Okay, so here's the photorespiratory pathway. And again, I don't want you to memorize anything about this. There's a couple important things that you should see. First of all, it involves 3 different organelles: chloroplast, peroxisomes, and mitochondria. What isn't obvious from this, but probably if you think about it makes sense, is all of these enzymatic steps that function in the photorespiratory pathway have not evolved to be part of the photorespiratory pathway. That would be a heck of a lot of specialized evolution. All of these enzymes serve other functions in the cell as well and have been, let's say, hijacked by photorespiration to carry out something that's going to, well, basically the important thing is dealing with the glycolate that's produced as a byproduct of the oxygenation reaction. So let's think about this from the perspective of what the cell could do to deal with that glycolate. If we look at aquatic algae, organisms that live surrounded by the water, they don't produce a whole lot of glycolate for reasons that we'll see in just a little while. But the glycolate that they do produce, they just release it into their environment. They get rid of a toxic compound by just excreting it, and the water carries it away. Right? So that's beneficial, and it gets rid of a toxic compound, but it's taking, for each glycolate, that's 2-carbon compound. That's 2 carbons that the organism has invested photosynthetic energy into fix, that it's throwing away. So that's not good. Well, if we think about terrestrial plants, first of all, they're not surrounded by water, so they can't excrete the glycolate. They have no way of getting rid of it. And second, giving away those carbons would seem to be wasteful. So what this whole photorespiratory pathway is trying to do is to recover the carbons in glycolate back into intermediates that the Calvin cycle can use. And it's done that using a whole bunch of enzymes that are there for other reasons. So just to give you a general picture of what's going on, I don't know how well you can see the colors. The black colors represent the movement of carbon atoms, and the red colors represent the movement of nitrogen atoms, and we'll see where nitrogen comes in here in just a second. Okay, so it's a complicated pathway, but the net result of this pathway is 2 molecules of glycolate that are produced from the oxygenation reaction of Rubisco. So that's a total of 4 carbons. Follow this whole pathway all the way around, and back up there, right under the Calvin cycle, you have 3-phosphoglycerate, PGA, a 3-carbon compound. So what is happening here, the net of what is happening here, is recovering 3 out of the 4 carbons that are in glycolate back into PGA. Remember, PGA is the first product that's produced by the carboxylation reaction. So it's going right into the Calvin cycle. So down here in the mitochondria is where the CO2 is lost, where that 1 carbon is lost. Okay, so we have uptake of oxygen, we have production of CO2, that's why it's called photorespiration. We're taking a toxic compound and recovering it back into compounds that are normally used in the Calvin cycle. But there's a cost to this. There's a cost of at least 2 ATP to do this. 1 ATP that's used directly to convert glycerate into phosphoglycerate, and another ATP that's used indirectly because down here, associated with this loss of CO2 reaction, there's also a release of ammonia. Nitrogen is fairly precious to plants, and they don't just get rid of this ammonia. They need to reassimilate it. We'll talk about nitrogen assimilation in a couple of lectures. But that reassimilation of the ammonia that's lost down here costs ATP as well. So this recycling of carbon, of 3 of the 4 carbons in 2 glycolate molecules, costs some energy as well. But what it allows the plant to do is a lot better than those algae that just excrete the glycolate into their medium. So, we can ask the question, does this pathway, is it a net benefit or a net cost to photosynthetic organisms, to plants? Is it a net benefit or a net cost? Well, let's add up some of the things, or at least list some of the things. In what way is photorespiration beneficial?
As you said, it saves saves 3 of the 4.
Okay, so it's recycling 3 of the 4 carbons. What else? What else is beneficial about this? Gets rid of the glycolate, right? Glycolate is toxic, right? So what is a cost to the organism here? How about Jonathan? What's the cost to this of running photorespiration? In what sense? Okay, so you've got ATP costs up here, and one of the things that you might notice is it's got NAD being used in a couple places here. Notice that in the mitochondria, NAD is being converted into NADH, and back here in the peroxisomes, it's going the other way. So there's no net effect on reductant, but there is a net effect in terms of ATP. One other cost. What's another cost that's in here? And so why is that a cost?
Because it's so hard to bind it in the first place.
There's a more expensive cost than that. Where did this carbon dioxide ultimately come from? Well, ultimately, in terms of the plant, it came from RUBP, that 5-carbon acceptor. And every one of those carbons in there cost 3 ATP and 2 NADPH to make, right? So giving away a CO2 down here is the equivalent of giving away 3 ATP and 2 NADPH. So there's a significant cost energetically here. So is this a benefit or a net benefit or net cost to the plant? No, I'm just talking about, let's compare a plant without photorespiration and a plant with photorespiration. We've got this list of things that are costs and benefits. Is it a net benefit? How do you know?
Well, I mean, you could always photosynthesize more, but if you have a buildup of toxin, that'd be different.
Okay. Yeah. And maybe an easier way to say it is, it's there. Evolution selected for it, right? There must be some benefit to it. We may not have quantified that benefit correctly, but the fact that it's there means there's some benefit to it. There's an even more compelling piece of evidence that this is important. And that comes from the fact, let's imagine that you could make a mutation in some enzyme in the pathway that's necessary for photorespiration that didn't otherwise have a big effect on the cell. As I said, all these enzymes play roles in other pathways, but let's just assume that you could mutate an enzyme so that photorespiration didn't work but had no other effects on the cell. Would this have a negative effect on the plants? Okay, why? Probably is kind of a wishy-washy answer. Let's be a little bit more.
You're asking that question.
So. Yeah. Yeah. You guys are starting to figure me out. Well, one thing we know for sure is glycolate could be a problem, right? If you can't get rid of the glycolate, that's going to be a problem. But it turns out there's a more important problem. Plants that can't do photorespiration are much more susceptible to light-induced damage. And we haven't talked much about this light-induced damage. We'll talk about it on Thursday. But it has to do with the fact that photorespiration is a sink for the products of the light reaction. Right? It's using up ATP and NADPH that are produced by the light reactions to make all of this go. If you put the plant under conditions where it's absorbing more light energy than it can use to drive photosynthesis, then we'll see that damage can happen to the plant. Photorespiration provides a new sink, an additional sink for those products of the light reaction. So Let's see, have to go all the way back here to the beginning. Remember this picture. I said how important it was. The coupling between the light reactions and the dark reactions by ATP and NADPH. The Calvin cycle is the main sink for these things, but photorespiration is another sink for ATP and NADPH. And under excess light conditions, photorespiration provides a mechanism of using these guys up so the light reactions can continue to happen. If they can't happen, if the plant's receiving more light energy than it can use, as far as using up the products of ATP and NADPH, then damage can happen. And we'll see what the characteristics of that damage is in the lecture on Thursday. Okay, so although what you will commonly hear from farmers, from a lot of people who think about agriculture, is that photorespiration is bad for plants. You should tell them, and you should be able to explain to them why that's wrong. That in fact, photorespiration, at least in the context that we've been talking about it, serves several beneficial roles for plants. And if we just blindly take photorespiration away, the plants will actually do worse.
So, is there an effort from plant breeders to get rid of photorespiration?
There has been, yep. There's been a lot of effort at sort of the molecular level to manipulate the relative binding affinities of oxygen and CO2. Because if you could get rid of oxygen binding, that would take care of a big problem here, right? Well, the government spent millions and millions of dollars to do this, and the outcome of that was we can't do it because the oxygen and CO2 binding sites are so tightly overlapping. If you change the oxygen binding site, you change the CO2 binding site as well. So you can't do that. And that should give you an important piece of information from an evolutionary perspective. Rubisco could not evolve to an enzyme where this competition between oxygen and CO2 was not a problem. Right? So what could evolution do? One of the things evolution did was that photorespiratory pathway, right? We got all this other stuff that deals with the products of photorespiration. Let's think about it from a larger perspective. What could a photosynthetic organism do to get around the problems of Rubisco? Well, it can't change the catalytic cycle. That's built into the enzyme. Can't really change the affinity for CO2 or the competition between oxygen and CO2 because that's built into the enzyme. If you change those drastically, the enzyme doesn't work anymore. But what else could a plant do? Anna? Say that again? Yes, okay, that gets around these two problems but does not get around this problem. You add more of them, it's just the same competition. What else?
We could have Rubisco in compartment where O2 doesn't.
Okay, so somehow keep oxygen away from Rubisco. Right? That's one possibility. What else? Should be another corresponding one. If oxygen and CO2 are competing, Anna's saying get rid of the oxygen. What else could you do to limit that competition?
Concentrating the CO2.
Yeah, right. Either concentrating the CO2 or getting rid of the oxygen or both. Right? And that's basically what's happened. The responses to Rubisco and photorespiration have been not changing Rubisco because clearly evolution hasn't been able to do that. Rubisco's been around for about 3.5 billion years. You'd think over that time that if it could have been altered by evolution, it would have been. Okay, so what we want to do is think about these mechanisms of either increasing CO2 or decreasing oxygen concentration. Let's just start here. Okay, so the mechanisms that have evolved to deal with this, they're basically three mechanisms. One of the simplest ones we're really not going to talk about. It's mentioned in the textbook. And that is active transport of CO2. Spend metabolic energy to pump CO2 into the cells or into the chloroplast where Rubisco is functioning. Right? Seems relatively straightforward. Interestingly, you only see that in aquatic algae and aquatic plants. So aquatic, they're angiosperms that live underwater. Many of those do active transport of CO2 into the cells and or into the chloroplast where Rubisco is functioning. But no terrestrial plants do that. Yeah, but isn't the CO2 that we're thinking about in the plants also ultimately dissolved in water? But the active transport is happening not from the atmosphere. It's happening from the liquid around the surface of the cell into the cell. Yeah, I mean, that's a good point. But people have argued about this for a long time. And there's not really a good explanation for why active transport of CO2 is limited to aquatic organisms.
Wait, I thought you said that each mesophyll cell is connected to an aerospace where they take out.
It is. But what surrounds the cell?
Water.
And where would the transporter that is taking CO2 from outside the cell to inside the cell be located? If there's a transport protein that moves CO2 from outside the cell to inside the cell, where will that protein be located? In the plasma membrane. So where does the CO2 that is being transported bind? From the air or from the water? Water. It's being transported into water. So the fact that it's in contact with airspace just means that there's a different way to get the air to the surface of the water on the cell. But the mechanism of transport into the cell is still from water to water. There's no transport of CO2 from the air directly into the cytoplasm, because the air is never directly in contact with the cytoplasm. There's always a film of water on the surface of the cells. Okay? Okay, so let's think about how terrestrial plants have gotten around this. And one of the main things that led people to understand that there was a different mechanism or a mechanism of getting around this problem of oxygenation was the fact that if we make a plot of rate of photosynthesis as a function of oxygen concentration, what we see is for the plants that we've been talking about that do the Calvin cycle, the C3 plants, it goes like this. Increasing oxygen concentration, decreasing rates of photosynthesis, at least as measured as CO2 fixation, net CO2 fixation. Right? No surprise. Higher oxygen, more oxygen competition, more carboxylation, more oxygenation and less carboxylation. But there's a whole group of plants that look like that. And these are referred to as C4 plants. Remember that C3 comes from the fact that the first product of photosynthesis that radioactively labeled carbon shows up in is a C3 compound, PGA, phosphoglyceric acid. We'll see that in C4 plants it shows up first in a different compound. Okay? So there are plants that are insensitive to oxygen. Another thing that turns out to be different is the anatomy of the leaf. Let's look at two different types of cells in a leaf. The mesophyll cells and the bundle sheath cells. The mesophyll cells are all these cells between the epidermal layers. So there's in this one there isn't really any palisade cells. There's mostly just spongy mesophyll, all these guys. And then these are the vascular bundles. And the bundle sheath cells are these cells that surround the vascular bundles. Okay, they're sort of like the boundary between the mesophyll cells and the vascular tissue. So if we look in a C3 plant, the mesophyll cells have chloroplast. The bundle sheath cells have no chloroplast. If you look in a C4 plant, these guys that show oxygen-insensitive photosynthesis, there are chloroplasts in both the bundle sheath and the mesophyll cells. And when you look at these bundle sheath cells in C4 plants, they're enlarged. And they called this Kranz anatomy. Do I have that up there? Yeah. Kranz anatomy. Anybody speak German in here? Yeah, so what's Kranz mean? It's a ring, yeah. Yeah, so this very distinctive ring of cells that shows up around the vascular tissues in C4 plants, but not in C3 plants. So there's some anatomical difference here that goes to the level of the fact that there are chloroplasts in both mesophyll and bundle sheath cells in C4 plants and not in C3 plants. But it goes a lot farther than that. And this is sort of a summary of the biochemistry of what's going on in the mesophyll cells versus the bundle sheath cells. And let's just take a quick look at this, and then we'll go back and look at the details of it. So in C3 plants, the chloroplasts have Rubisco and the Calvin cycle. Obviously, if there's no, in the bundle sheath, there's no chloroplast, so it's not there. In C4 plants, Rubisco and the Calvin cycle is only in the bundle sheath cells. In the mesophyll cells, there's no Rubisco and no Calvin cycle. Seems bizarre, doesn't it? Until you understand what's going on here, it seems completely crazy. So let's go through and let's make some sense out of this. So first thing we want to do is think about why we call these guys C4 plants. They're called C4 plants because up here at the top there's our CO2. And remember, have I talked about CO2 and bicarbonate equilibria? Did I mention that? I should do that. If we take CO2 that's dissolved in water, plus water, forms a molecule called carbonic acid. Carbonic acid. Carbonic acid can dissociate to form bicarbonate and a proton. And it turns out that in fresh water, and even in the cytoplasm, there's a lot more bicarbonate than there is dissolved CO2. The equilibrium favors the formation of bicarbonate. But the equilibrium is relatively slow between those guys. And there's an enzyme that catalyzes this reaction called carbonic anhydrase. It's just speeding up the equilibration between CO2 and bicarbonate. So in the cytoplasm, there's an equilibrium between CO2 and bicarbonate. So if we add radioactively labeled CO2 very quickly a lot of that radioactively labeled carbon is in bicarbonate. And in C4 plants, the initial reaction that fixes carbon dioxide does not involve Rubisco. It involves an enzyme called phosphoenolpyruvate carboxylase, PEP carboxylase. And it converts a 3-carbon compound, adds a CO2 to it, or adds a bicarbonate to it, producing a 4-carbon compound. So Rubisco is not the initial enzyme that's involved in CO2 fixation in C4 plants. But we said Rubisco's still there, it's just in a different cell. Remember, these are the mesophyll cells. These are the bundle sheath cells. Okay, so the question is, how does Rubisco still play a role in this? And it's relatively straightforward. The C4 acid that's produced in the bundle sheath cells diffuses through the plasmodesmata, it's produced in the mesophyll cells, diffuses into the bundle sheath cells. In the bundle sheath cells, that C4 acid is decarboxylated, releasing CO2 and a 3-carbon acid, pyruvate, which then diffuses back into the mesophyll cell and is converted back into PEP, the substrate for the initial reaction. So what's the net of what's going on here? What's the net that's going on here? We start off with CO2 that's being fixed in the mesophyll cells, and we end up with CO2 that's released in the bundle sheath cells. Think back to what we were talking about in terms of how can evolution find a way to get around the problems of Rubisco. What could this accomplish?
There's more CO2 where Rubisco is?
Yeah, if the concentration of CO2 is higher here than it is here. Right? And that's what's going on here. This is nothing more than an elaborate metabolic pump. It's not a pump like active transport. There's not a protein in the membrane of these cells that's actively transporting CO2 in. There's exchange of 4-carbon and 3-carbon acids between these two cells, the net of which is the CO2 concentration in the bundle sheath cells is higher. Is there a cost to this? Close your eyes. Don't look at the reactions. If we're concentrating CO2 in the bundle sheath cells, is there a cost to this? There has to be, right? You're moving CO2 against the concentration gradient. You can't do that without investing energy. We know that from back in the first lectures we talked about. So associated with this pathway, there must be an additional cost in energy above the normal cost of the Calvin cycle. So we've got 3 ATPs and 2 NADPHs needed down here for the Calvin cycle. There has to be an additional cost, and I think I have a slide, that shows that additional cost. That additional cost is up here in the process that converts the 3-carbon acid pyruvate that comes back into the mesophyll cells, that converts it into phosphoenolpyruvate. There's 2 ATPs required there. So there's an additional 2 ATPs that are needed for C4 photosynthesis beyond what is normally needed for the Calvin cycle. This NADPH, yeah, so if we look at this, NADPH is used to convert the product of that first reaction to reduce it to another compound. It's reducing oxaloacetate to malate. But when you do the decarboxylation reaction in the bundle sheath cell, oops, the textbook's got it backwards. This is correct. You're using an NADPH here. This is incorrect. It's going the other way. It's converting NADP to NADPH. So there's no net use of reductant there. The only additional energy comes from this requirement for ATP. So if we think about it, this is accomplishing just what we wanted. It's a metabolic pump which is increasing the concentration of CO2 in those cells where Rubisco is. Just what we want to do. So the other part of the story that we mentioned was getting rid of oxygen. And I'm not going to go into a lot of details about this, but the chloroplasts in the mesophyll cells are normal chloroplasts. They have both photosystem 1, photosystem 2. They produce ATP and NADPH. In the bundle sheath cells, there's no photosystem 2. Only photosystem 1 in the chloroplasts there. What's the benefit of that as far as, what's the benefit of getting rid of photosystem 2? Francesca? So what? Okay, so there's no oxygen production in these chloroplasts. So why is there just photosystem 1? We didn't talk about this. What can you imagine would be, considering the overall picture here, what can you imagine would be the role that photosystem 1 would play in the bundle sheath cells? Okay, so if there's no oxygen, no photosystem 2, where are the electrons going to come from to reduce NADP to NADPH? Right, because the electrons that ultimately go to reduce NADP to NADPH come from oxygen. Without photosystem 2, there's no source of electrons there. Go ahead. Go ahead. Stephen. Yeah, sorry.
Wait, don't you use ATP?
Yeah. Do you need extra ATP? Do we need any ATP? Yes.
Yeah.
Right? So the one thing that we didn't talk about, but this becomes relevant now, is that there's another type of electron transport. The one that we talked about in lecture on the previous lecture was linear electron transport, where we have electrons that go from water to NADP. That uses both PS2 plus photosystem 1 and makes ATP and NADPH. But there's another one that uses just photosystem 1. And if it just has photosystem 1, there's no source of electrons. It can't do any net oxidation or reduction. But what it does is a cyclic pattern of electrons around photosystem 1. So the electrons that come out of photosystem 1 that normally would go to NADPH come back into photosystem 1. And this can make ATP. So it's called cyclic electron transport. So there's one of the supplementary study questions that asks you to think about, okay, the chloroplasts in the mesophyll cells make ATP and NADPH. The chloroplasts in the bundle sheath cells just make ATP. We know from looking at this picture that we need ATP and NADPH in the mesophyll cells. And if we have the Calvin cycle in the bundle sheath cells, then we need ATP and NADPH here. Where's ATP being made? Where's NADPH being made? Where are they being used? What must be transported? The question is really asking you to think about what's being transported between the two cells besides 4-carbon and 3-carbon molecules that are transporting the CO2 in. It's not the picture here does not come close to the complications of transport that are involved in this. And thinking about ATP and NADPH gives you some idea that there's a lot more going on here. Hannah. Can't be transported across the chloroplast envelope, right? But so, for example, they can move from the cytoplasm. There's plenty of ATP in the cytoplasm, and it can move through the plasmodesma from one cell to the next. NADPH the same way. So yeah, they can exchange from cytoplasm to cytoplasm, but they can't exchange directly from chloroplast to cytoplasm or chloroplast to chloroplast. Okay, so the net picture that you should take away from this is that what C4 photosynthesis has done has not replaced Rubisco. Calvin cycle's still here. But it's produced a spatial pump, 2 different cells, the net result of which is increasing the concentration of carbon dioxide in the site where Rubisco functions and decreasing the concentration of oxygen to the point where, under physiological conditions, there's no oxygenation reaction. There's no photorespiration necessary in a C4 plant. So let's come back to a question that we talked about in several lectures ago that had to do with the fact that C3 plants need to have their stomates open in order to do photosynthesis. We haven't talked about it, but one of the things that we know about C4 plants is their stomates can be mostly closed, and they can do normal photosynthesis. So thinking only about Fick's law and assume that the resistance to CO2 diffusing is the same in the epidermis of C3 and C4 plants. What's the only thing that could explain why C3 plants need to have their stomates open and C4 plants don't need to have their stomates open in order to do photosynthesis? Forget all the biochemistry. Just think about Fick's law, right? Just forget your biologist and think like a chemist for a minute. The fusion coefficient, we said, is the same. What's the only thing that can drive CO2 transport, CO2 diffusion across closed stomates?
You reduce the concentration inside.
Concentration of what?
CO2, so that they'll diffuse more.
Why does it diffuse more? You're correct, but you're missing an important word here.
The concentration gradient.
Yeah, the concentration gradient. So how could you make the concentration gradient bigger in a C4 plant than in a C3 plant?
You remove the CO2 from whatever, with the mesophyll cell.
Yeah, and so what does that mean about the characteristic of the enzyme that takes up CO2 in C4 photosynthesis versus Rubisco in C3 photosynthesis?
That end of it is better than Rubisco, because I think everything is better than Rubisco.
Everything, okay, but better in what way? Better in what way?
It's better, has more affinity.
Has higher affinity for CO2. That's it.
And faster filling?
It's also faster too. But faster will help in terms of, make it harder to keep up with things. But it's the higher affinity that ultimately makes the difference. So PEP carboxylase can lower the internal CO2 concentration in the cells and inside the leaf much more than Rubisco can. This has 1,000 times higher affinity for CO2 than Rubisco does. That's why C4 plants can do photosynthesis with their stomates mostly closed, while C3 plants can't. Because Rubisco has a crummy enzyme. It can't lower that CO2 concentration very much. It can't make a big gradient. So the stomates have to be open. You have to have a low resistance pathway. But in C4 plants, because it can't make a big gradient, you can close the stomates a lot more and still do the same amount of photosynthesis. So why is that important?
Water.
What about water?
If the stomates are closed, there's less water loss?
And is that good for the plant?
Yeah.
Yeah, it can be. So in terms of water use efficiency, C4 plants can do a lot more photosynthesis per water lost than C3 plants can. Is that going to have ecological significance? It's going to help determine where C4 plants grow rather than C3 plants. Yeah, of course it will. There's one other important difference.
Oh, I have a question.
Okay, sorry, go ahead.
So, having this extra process of C4 plants, it requires some energy too, right? So,
Yep.
if water is not limiting, then C4 plants don't really have an advantage over.
Good question. Yeah. So keep that in mind because we'll come back to that next lecture. Because one of the things we'll talk about next lecture is the physiological consequences of competition between, and physiological and ecological consequences of competition between C3 and C4. Because it should be clear, C4 plants have not replaced C3 plants, right? So one of the things that you should be looking for in your readings for the lecture on Tuesday is what are the conditions under which C3 plants are going to have an advantage, right? I'll give you a clue in that in just a little bit, but the answer is going to be interesting. Okay, one other thing that differentiates C4 from C3 plants. Well, one of many other things, but one that's important from an agricultural perspective. C3 plants have got Rubisco in all their mesophyll cells. Where's Rubisco in C4 plants? Bundle sheath. And which one are there more of, mesophyll or bundle sheath? Lots more mesophyll cells. So which plant has less Rubisco? C3 or C4? C4. So which plant needs less nitrogen? C4, because remember, Rubisco is a protein, most plentiful protein on Earth. If you can get away with less Rubisco, it makes the plant more nitrogen use efficient. So it's even more complicated. It's not just in terms of photosynthesis and not just in terms of water and not just in terms of nitrogen. There's lots of advantages that C4 plants have over C3 plants. But again, clearly C4 plants have not taken over the Earth. Okay, one more quick thing about C4 versus C3. About 350 million years ago, the carbon dioxide concentration in the atmosphere dropped significantly. Where did all that CO2 go? Nope. Anybody geologist here? No geologists? You're standing on top of it. There's 3 miles of limestone underneath our feet. What's limestone? Carbonate, right? So the CO2 in the atmosphere was largely taken up by single-celled photosynthetic organisms that also made their shells, their cell walls, out of carbonate. And it's all those cell walls that are now in, not just in central New York, but all over the world, big deposits of limestone. So the CO2 concentration in the atmosphere, because of the proliferation of these photosynthetic algae, the CO2 concentration dropped significantly. So Rubisco evolved in an environment where there was much higher CO2. And it's only about 350 million years ago that the CO2 concentration dropped precipitously. So you might expect, when did C4 plants evolve? 350 million years ago, approximately. Interesting thing is that are C4 plants, in terms of C4 photosynthesis, monophyletic? That is, are they all derived from a single kind of plant? And the answer is no. C4 plants, I can't remember what the latest number is. C4 plants, C4 photosynthesis has evolved independently at least 18 different times.
And are all the pathways the same?
Good question. What would you expect? If it evolved 18 different times, would you expect them all to be identical?
No.
No, they're not. And the way they differ, one of the main ways they differ, is what are the C3 and C4 acids that are exchanged between the mesophyll and bundle sheath cells. Some cases it's malate and pyruvate. Other times it's, I can't even remember what they are. But there's a number of different variations on this theme that represent the fact that this evolved independently a number of different times. One of the things we didn't talk about that's important, this enzyme, PEP carboxylase, did this evolve with C4 plants? You don't know the answer to this. No. We'll see when we talk about respiration that PEP carboxylase is a key enzyme in carbon metabolism associated with the citric acid cycle. So C4 photosynthesis is very much like photorespiration. It borrowed existing enzymes to carry out different functions. None of these enzymes that catalyze this metabolic CO2 pump are new enzymes. They were already there. Certainly they must be regulated differently. For example, PEP carboxylase. It's carrying out the same reaction, but the conditions under which it carries it out is quite different in C4 photosynthesis versus the role it normally plays in respiration. Where it's located in the PEP carboxylase that functions in respiration is in mitochondria, not in chloroplasts. But in C4 plants, there's PEP carboxylase in the chloroplasts, at least in the mesophyll cells. So it's a very interesting evolutionary and cell biology component of this that we're not even going to get into. But what this represents is a very widespread response to lowering the CO2, Rubisco suddenly becoming to be a real problem for plants. And this is one response to it, one mechanism of getting around it. Okay, so questions on this before we move on? Keep in mind that what I want you to be thinking about is not the details of the enzymes that are involved here, other than PEP carboxylase. You don't need to worry about any of the other ones. I don't even care if you remember what the names of the acids that move back and forth are, but you should know that what's coming from mesophyll to bundle sheath is a C4 acid, and what's going back is a C3 acid, and the extra carbon shows up as CO2 to increase the carbon dioxide concentration. That part, that's the big picture. That's what I want you to know. Yes?
For photosystem 2, where do you were talking about how the electrons are cycling through. Where do the electrons come from?
For photosystem 2? Photosystem 1? This, yeah, for the cyclic electron flow? Yeah. Okay, so I'll take a second to talk about that. So let's just take our reaction center, P700 of photosystem 1. Right? So we know that what happens in light, if these are electron donors up here and electron acceptors down here, what light energy does is creates a really good electron donor. And what normally happens is these electrons go through a bunch of alphabet soup, right? And then you get down here to NADP, getting converted to NADPH. And normally, the electrons that are coming in here come from photosystem 2, right? In the bundle sheath cells, there's no photosystem 2. So what happens is the electrons that come out of photosystem 1 basically cycle back around. So it's just an electron. Photosystem 1 gives up an electron, it travels around through a bunch of carriers and comes back to photosystem 1. But if this can make ATP, if this cyclic process of electron transport can make ATP, what else must be happening in this electron transport? Well, of course, there's energy loss. Proton transport, right? So somehow this pattern of electron transport must also move protons across the membrane, because that's how ATP is made. Yeah, it does. It goes back through the cytochrome complex. The gory details of this are not necessary. I mean, I could spend a semester talking about all the details of electron transfer associated with linear and cyclic electron transport. I really don't want you to spend a lot of time on this. The key thing, this could have been a good exam question. If there's a pattern of cyclic electron transport around photosystem 1, what else must it do besides move electrons around? If it makes ATP, it's got to pump protons too. And it turns out the proton pumping involves the same stuff that is between photosystem 2 and photosystem 1 as linear electron transport does. Okay? You don't need to know that. I'm happy to talk about it, but you don't need to know that. Other questions about?
It doesn't solve the question of where does it come from, or is it just there?
No, it's an electron. It starts as an electron from the reaction center pigment. Remember what we said, an excited state? So if you've got a pair of electrons in an orbital, when it absorbs light energy, that electron gets kicked up to a more distant orbital. This is the electron that goes away. When this gives up an electron, that electron goes away. And all you're doing is passing it around and giving it back to P700, but you're giving it back to the ground state rather than to the excited state. So the energy of that excited state has been used to drive this pattern. Okay? But there's no net oxidation or reduction. The place where the electron starts is the place where the electron stops. Okay? All right. So let's finish up by talking about another way that plants can show this characteristic of oxygen sensitivity. So we said that C3 plants look like this. We said that C4 plants look like this. And there's another group of plants that have the same characteristic. And these, so pretty much the same thing as C4. And these are called CAM plants. CAM stands for crassulacean I'm terrible at spelling. I can't remember. Crassulaceae are a group of plants, mostly succulents, things like a lot of cactus. And they're called crassulacean acid metabolism plants because somewhere back, probably some poor person out in the desert who was looking for water discovered that when they tried to get water out of a cactus plant, just basically, squeeze it out to drink it. In the daytime, it doesn't taste very good, but in the night, it tastes very acidic. Then in the nighttime, the water in the cactus plants is acidic. And then biologists came along and measured it and stuff. And it turns out that the water in the cactus plant gets very acidic at night. Okay, so the question is, why? This crassulacean acid metabolism is related to that. Simon?
Just a side note, crassula is not a cactus.
Oh.
Isn't the crassulaceae, and crassula also has, the family has palincoe, plancho is a common plant.
So, but succulents is...
Yeah, succulents in general do this generally. I think it was just discovered in a particular crassulaceous, not a cactaceous plant.
Okay,
But
so we have a real botanist, and that's important. We don't want to give the wrong perspective here. Okay, but I think one of the things that you should take away from this right from the beginning is that this is limited mostly to plants that live in environments where water is limiting. That in fact, this whole behavior that makes CAM plants be oxygen independent has nothing to do with photorespiration, but has everything to do with trying to conserve water. So keep that in mind as we go through discussing the physiology of CAM plants. Okay. Yeah, let's skip that and that. We'll come back to that on Tuesday. So what we want to do is think about what's happening in the CAM plants that allows them to be two things: oxygen insensitive and be able to conserve water. So we saw that C4 plants are conserving water, but CAM plants do it in a very different way. CAM plants have a reversed cycle of stomatal opening and closing. In C4 and C3 plants, the stomates are open in the day and closed at night. CAM plants, they're open at night and closed in the day. And what the challenge is to figure out what open at night and closed at day, how it allows the plant to do photosynthesis. Okay, so in terms of water loss, go back a couple lectures. What's the advantage of opening your stomates at night rather than in the daytime? Less loss of water, but why? But why would there be less at night than in the daytime? Okay, so what's the driving force for evaporation? Why is there more evaporation in the daytime than in the night? It's warmer. That's basically it, right? Because remember, the relative humidity is temperature dependent. The cooler it is, the higher the relative humidity for the same water content. So at night, there's a much lower gradient for water loss. So opening stomates at night has much lower consequences for a plant than opening in the daytime. That's important. But the light reactions can only happen in the daytime. You can only make ATP and NADPH in the daytime. So if the CO2 is coming in at night, but the light reactions are going on in the daytime, it's not going to help unless it can do something to couple them together. And the way that happens is that at night, carbon dioxide enters the leaf. And using the same enzyme that C4 plants use, PEP carboxylase, that CO2 is fixed into acids that then are stored in the vacuole. So the fact that these plants taste acidic at night is because these vacuoles are chock full of typically malic acid. Where's the energy to do this coming from? It costs energy to do this. That energy is coming from starch that's stored in the chloroplasts in the daytime. So starch is being metabolized to provide the energy to drive this reaction at night. Now it's not so hard to understand what's going to happen in the daytime. The daytime, the stomates are closed. The malic acid is released from the vacuole. It's decarboxylated, just like in the C3 plant or in the C4 plants, and fixed via the Calvin cycle. And some of this carbon has to be socked away at night as starch to regenerate the starch that was used up the night before, and the rest, the extra stuff, goes on to be used outside the plant. So while the C4 system is a spatial pump, two different cells with metabolism going on simultaneously, CAM photosynthesis is a temporal pump. At night, the CO2 is fixed and stored in the vacuole as malic acid. In the daytime, the stomates are closed, the malic acid is released, decarboxylated in the chloroplast, and then fixed by the Calvin cycle. So where do the costs associated with CAM photosynthesis come in? It's not shown on here. I mean, the reactions are shown on here, but the cost is not shown on here. Where's some place that energy is going to be required here? Conversion, yeah. So there's some energy that's required in the production of PEP. But there's one more place where it's even more important. Yeah, you use it up here, but you get it back over here. How about here? Does it cost energy to accumulate malic acid in the vacuole? Can you just keep putting malic acid in the vacuole for free? No. So there's a cost here in terms of storing the malic acid. There's got to be an ATP-dependent transporter, ultimately, that is putting that malic acid into the vacuole. Grace, you look confused.
No, I was just wondering, what about storing the starch? Isn't that also a problem?
Is it a problem to store starch?
Yeah, I think you have to use energy to get it into the chloroplast after you...
To go from triose phosphate to starch?
Oh, okay, so it's all happening inside the chloroplast.
All happening inside the... Yeah, starch in plants is stored in the chloroplast. In us, it's not. But in plants, starch is stored in the chloroplast. Okay, so are we okay with this? The difference between what's going on in C4 and CAM plants? Yes?
At night, the CO2 comes from the atmosphere also from respiration.
There's also respiration that can contribute to that, right?
How much CO2 from respiration
Um,
compared to from air? Can you say how much?
well, if a plant is going to grow, what fraction of the CO2, let's say a plant is growing a lot. It turns out that many of these plants that do CAM are quite efficient overall in their growth. So if a plant is growing a lot, could all the CO2 be coming from respiration? If it was all coming from respiration, would the plant grow? Couldn't. Right? If it just took fixed CO2 in the daytime and used the same CO2 at night, there's no net gain in carbon. Right? So most of it is coming from the atmosphere. Some is coming from respiration, but most of it is coming from the atmosphere. That's a good question. So under the worst case scenario, if it completely recycled its respiratory CO2, it would be able to sort of maintain itself, but it would never be able to grow. To grow, you got to add more carbon, right? Cells are mostly carbon, right? So you got to add more carbon to get them to grow. So it's got to have a net uptake of CO2. Okay, let's finish up then by just saying something briefly about this process. How is starch made in chloroplasts, and why is making starch in chloroplasts important? Obviously, in a CAM plant, making starch is important because this starch that's stored away in the daytime becomes the substrate to make phosphoenolpyruvate, the 3-carbon acceptor molecule that takes up the CO2. So in one sense, we can think in a CAM plant that a lot of the carbon is just sort of sloshing back and forth between it's stored away at starch at night, gets converted into malate, and in the daytime, it goes in the opposite direction. You have a pool of carbon that's just moving back and forth. You have another pool of carbon where there's net carbon uptake at night and carbon that goes in to make more plant in the daytime. But all plants produce starch at night, and we need to think about how that happens and why it happens. So let's just finish up by talking about this in the context of the larger picture of how carbon moves around in plants. So if we think about in the daytime, the plant's producing through photosynthesis triose-phosphates. Those triose-phosphates do not accumulate in the chloroplast. If they accumulate in the chloroplast, the first thing that happens is the higher concentration of triose-phosphate feedback inhibits the Calvin cycle, and photosynthesis stops. Triose-phosphates don't accumulate. In the chloroplast, they can be used to make starch, or as we'll see when we'll talk about this on next Tuesday, they can be exported from the chloroplast into the cytoplasm, where they can be used to make sucrose, to be exported, or it doesn't have it in here, they can also be used for respiration in the cells. So there's a number of different sinks that are going on in the daytime. Sink for making starch, a sink for making sucrose, and a sink for using respiration. How about at night? What's the supply of triose-phosphates at night? Is there any photosynthesis at night? No. So the only place that in the chloroplast that triose-phosphates could come from is from starch that's stored away in the chloroplast in the daytime. In fact, much of the energy that the mesophyll cells in the leaf need to do their normal things at night, that energy comes from carbon that's stored as starch. Okay, so we need to think about starch synthesis in the chloroplast is serving an important role of socking away some carbon in the daytime to supply the cell at night. Okay, so one of the important things that starch metabolism has to do is save energy in the chloroplast that the chloroplast and the mesophyll cell can use at night when photosynthesis isn't available. Another important part about this is, that sucrose that's produced in the mesophyll cells is distributed to the whole plant. Does this process of taking sucrose out of the mesophyll cells and sharing it with the rest of the plant only happen in the daytime? We haven't talked about phloem transport yet, but it doesn't. It happens at night as well. Where's the sucrose coming from at night? It's ultimately coming from starch. So it's not just the mesophyll cells, it's the whole plant. that the chloroplasts in the mesophyll are storing starch away for. Okay. So that's one of the important things. That's probably the most important thing. But there's another thing that is important as well, and that is that starch can basically function starch synthesis has an overflow mechanism. So when photosynthesis is producing more triose than all these things can use up. If these things sort of backed up, then triose start to accumulate in the chloroplast and photosynthesis would stop. Under those conditions, the plant makes more starch in the chloroplast than it needs at night. Starch becomes a temporary sink for the products of photosynthesis. So starch synthesis is a sink for the products, for triose phosphate, really. So when triose phosphate accumulates in the chloroplast in the daytime, because these sinks become limited, the synthesis of starch can be turned on. And I think the study question for this lecture topic is asking you to consider the balance between those two. The requirement that all plants require starch to be made and stored in the chloroplast in the daytime to support processes at night. But this overflow mechanism is also there. And it's asking you to think about the regulation of those two different processes. Okay, let's stop there and we'll finish up next time talking just briefly about starch and sucrose synthesis. And then we'll move on to the last lecture on photosynthesis, which is related to sort of physiology and ecology from the big picture, in the big picture. So, for example, why do C3 and C4 plants coexist? You know, that's a very important question from whether you're a farmer or whether you're an ecologist. And we'll put together all the things that we've been talking about over the last 3 lectures to be able to answer that question.